Hello, young scientists. Today, your mission is DNA extraction. To help you on your mission, the Biotech Club will be your support team. My name is Samiha Sayed, and I am president of Biotech Club. Jillian Smith is our vice president of event planning. Zohair Ullah Sayed is our vice president of community relations. Ikram Isa is our chair of recruitment. And Dr. Persky is our advisor and favorite scientist. All right, time for questions. Does anyone have any questions?
What is DNA?
DNA is like instructions for our body, like a blueprint that tells our body to develop, grow, and function. DNA is really, really small, but even though it's super small, it has a shape called a double helix, which looks like a braid made of hair. DNA is short for deoxyribonucleic acid. So what is DNA made of? It's probably a question you're thinking right now. So DNA is a long, thin molecule made of something called nucleotides. There are 4 different types of nucleotides: adenine, thymine, cytosine, and guanine. They are usually represented by their first letter. So for adenine, that's A, for thymine, that's T, for cytosine, that's C, and for guanine, that's G.
What is extraction?
All right, so extraction means taking something out of something. In this case, we are taking DNA out of strawberries and bananas using a special liquid called extraction liquid. Also, in case you're wondering, cheesecloth is a word that you'll hear as well. So that just means a woven cloth made of cotton. So typically cheesecloths are used for wrapping cheese. That's why it's called cheesecloth. And if you don't have a cheesecloth, you could use other cloths or filters for this purpose.
So for this experiment, you're going to need a couple of items. The first thing you'll need is a measuring cup. Next, you'll need a measuring spoon, some rubbing alcohol, half a teaspoon of salt, a 1/3 cup of water. You will need 1 tablespoon Dawn dishwashing detergent or a comparable brand, and shampoo works as well. You'll also need a glass or a small bowl, a cheesecloth as Samiha mentioned. You'll also need a funnel, a tall drinking glass or beaker, half of a banana or 3 fresh thawed strawberries, minus the green tops as well. You'll also need a test tube or a small glass jar. Baby food or spice jars work as well. And then lastly, you'll need a long toothpick or skewer sticks.
All right, everyone, you've heard a lot about what we're doing. You heard what you need. I hope you took a moment, paused the video, went and grabbed all the supplies you would need, and you're ready to start this experiment. All right, everyone, so let's get started. So grab your measuring cup and you're gonna put in a 1/3 of a cup of water. So pause the video, go put in your 1/3 cup of water and come right back. All right, from here, make sure you don't get your clothes in anything, because then your clothes will get all messy. So pull up your sleeves if you're wearing long sleeves. All right, so take your 1 tablespoon measuring spoon, and you're gonna measure out with your Dawn dish liquid. You're gonna measure out 1 tablespoon. Make sure you get it all the way to the brim. And brink. All right, put that in. You can give it a little swirl if you want to. Pretty fun. You don't want to whisk it though, because it's going to make it all foamy, and you don't want it to be foamy. It's not bad if it is a little foamy, but less foam is better in this one. And then you're going to need salt. So everyone grab your salt. Mine looks like this. You can get it at the grocery store for pennies. It's very, very inexpensive. It's great. And I like to buy the big one because I really like salt. In moderation, it's good for you. Not in moderation, it's bad for you. So take your salt in moderation. All right, so you have your half a teaspoon of salt, and you're gonna put that in your mixture, and you're gonna give it just a gentle stir until your whole water, it looks a little blue if you're using colored dish liquid. It may not turn blue if you're using shampoo. Okay, so tap that off, make sure you're not dripping across your clean station, and you're just gonna set it aside. Okay, so check before this next part that you put your alcohol, mine's all frosty because it's been in the freezer. So pause the video, go run, and put it into your freezer. Don't worry, it won't freeze solid because of the alcohol percentage. If there's a high enough alcohol percentage, it will not freeze. You could leave it in there for weeks. Mine's been in there for about a week because I forgot about it. It won't explode. It would be totally fine. So go throw it in the freezer and come on back. All right. Next, we need a plastic bag. So I picked kind of a bigger plastic bag because I want to have enough space to do a lot of mashing. So I don't know how many of you have ever made slime, but slime is really cool because you can mush it, you can stretch it, you can play with it for a long time, and it doesn't ever like disintegrate or fall apart. If you did it for a couple months, it probably would. All right, so I'm gonna do a half a banana. So I cut my banana in half, and I don't want the peel. So I'm going to take the peel off. And I'm gonna set that aside to throw away later. All right, so put that banana in your bag. Woo, and I have banana on my fingers now. Ooh, ooh, ooh. And you're gonna squish out all the air. So I'm gonna roll it to make sure I don't have any air in this bag. Squish, 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 squish. And you're gonna seal it. Make sure if you're using a double seal, you seal both of the seals. Okay, so it should look like this. Very, very skinny, all the air squished out. And now you're gonna play with it like it's slime. So squish it, mush it. What we're doing right now is inside a cell is what is called DNA. And Samia covered this when we first got started. What is DNA? And we discovered that it's a really special double helical structure that contains the code that makes you you. And every person has different codes, different sequences, and it is really what makes you uniquely you. So what we're doing with this banana is each of those cells has a protective coating on it. And in order to get the DNA out, you have to break down those cell walls. Oop, keep mashing. I almost stopped mashing because I started talking with my hands. So keep mashing. We're gonna break down all of those cell walls. So rub, 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 rub. Awesome, keep going just a little bit more. We have a couple more seconds. Break down all of those cells walls, because the more we break down those cell walls, the more DNA we can get out. I don't know about you, but I'm kind of curious what kind of DNA this banana has. All right, squish, squish, squish. Okay, so I'm gonna leave this for just a second, and I'm gonna go grab my filter, so my funnel and my cheesecloth or strainer, and I kind of made a makeshift one here. So this strainer, mine looks like this. I think it's so you can drain like a can of like artichoke hearts or something like that. And you put it on top of your funnel, or you put your cheesecloth inside. I did try a coffee filter once, but it's a lot easier to do it this way. You'll get a stronger yield from it. Okay, and then it's all gonna drip down into this little jar here. We want to get as much of the liquid without the pulp, which is the chunkier stuff of the banana. Wanna make sure we get a pretty clear yield. All right, it's time to add that mixture that we set aside at the very beginning. So your banana or your strawberries should look like this consistency. Kind of looks like baby food, looks a little scrunchy. Anyway, so grab your mixture. Look, mine's all blue. Yours should be blue too if you use blue detergent. All right, so open up that bag of banana, but don't lay it on the table, because then it will all spill out and you'll have less DNA. So go back and grab your 1 tablespoon measuring spoon. Okay, you're gonna take 3. Everyone show me 3 fingers. 3 fingers, good job. 3 tablespoons of this mixture, okay? It may seem a little thick, but that is okay. And you're gonna go 1 and 2 and 3. Good job. Good job. All right, and go ahead and set that aside. We're done with that for now. All right, so we're back to the mashing. Just like with slime, we have another about minute of mashing because you want to get all of that dish detergent and salt to work through, we'll call it the banana puree, okay? You want to make sure that it is working all through the mash of that banana. It'll get a little foamy, but that's okay. All right, and let me think. Why do you think we added salt to this? So let's start with the soap. I'm more curious about the soap first. So soap molecules and the mashing are helping to break the membranes of the cells and the nucleus to free that DNA. So the dish soap is now helping with all of the mashing we just did. It's helping to break down those cell walls. And the salt helps set the DNA free from the proteins that it's wrapped around. So proteins are what help it make that double helical structure that Miss Amia talked about at the very beginning. She said it was like almost like hair-like structure. So that salt is helping the DNA break free from that protein that's helping it be all corkscrewy. It's pretty cool. And the next step, we're going to add our rubbing alcohol. So your rubbing alcohol should be right here. All right, but first we need to strain it. That rubbing alcohol, I'll tell you what that does next. All right, so open up your bag. It should, I should show you guys this. It should look pretty similar to last time. It may be a little more foamy and you may be able to see thicker chunks. That's what we're trying to strain out of this. So open it up. So much puree. All right, so you're gonna pour it in. Let's see, can you all see that? Oop, there we go. That's better. Oh my goodness, look at all of that. All that clear yield that we are getting. Ooh. All righty, and strain it. All right, and we're just gonna let that sit. Sometimes it takes like 2 minutes. Sometimes it takes an hour. So that's why you want to really work through the banana and mash it up. And once you add the detergent, continue to mix and mash all of that, because the more liquid that comes down from this, the more yield you'll have, and then we can separate for the DNA. All right, so this I'm gonna let sit for a little bit, but I actually already did this experiment earlier, so I have one that is already done. Okay, so I'm gonna set this one aside, but you keep yours and you just wait a little bit. Wait a little bit more. Try to explain to someone else what you are learning in this video. Okay, so this is what mine looks like. It's been sitting for a while. As you know, bananas, when you let them sit, they turn brown, which is not as exciting. And I'm not gonna lie, I don't really like brown bananas, which is a little sad. Okay, so this rubbing alcohol, after you've strained it, you can discard the cheesecloth or the strainer or the pulp that's inside that. We're done with that. We just want the clear liquid at the end. All right, so you're gonna tilt your jar. So don't just dump it in like this. If you dump it in like this, you're going to have all sorts of mixed up DNA, pulp. It's not gonna turn out right, and your experiment won't work. Also, if your alcohol is not chilled, it won't work either, okay? Well, it'll kind of work, but it just won't work as well, and you won't get as much DNA out. Okay, so you're gonna tilt your jar like this, and you're gonna wanna pour your alcohol against the wall. So just very gently. There we go. All right, here we go. And I poured in a lot so we can see it separate. Okay, so wait. All right, I don't know if you guys can see this, but on top of that dark brown color, you can see little bits of clear DNA coming up. So in order to pull out the DNA, that's what your skewer is for. Everyone show me your skewer. Awesome. Okay, so set your rubbing alcohol aside, put the lid on it, because if you spill it on like a table, it can take off a finish on a table. So you want to make sure you cap that pretty quickly. Kind of like nail polish remover. You don't want to dump that on your mom's end table. Okay, so see there? You can see the bits of DNA. Let me try to get it closer. You see all that white stuff? Okay, that white stuff is what we're looking for. That's the DNA. So you're gonna take your skewer and you're gonna try to spool it. But if you stick it into the brown stuff, it will start to pull up things that are not DNA. You don't want that. You only want the DNA. And so what happened was this alcohol precipitates the DNA, so it just turns the DNA into a solid that we can see. Very interesting. All right, so dig in that jar. I'm gonna set mine down so I can dig into it with you guys. Okay, I'm just gonna pick out the very top of the DNA. All right. Let me see if I can show it to you. Okay, I don't know. Let me try to grab one more. Okay. You will see a almost like jelly, like clear. Let's see, can you guys see that? Okay, you see that little jelly clear thing right here on the end? That is your DNA. Well, not your DNA. It's the strawberry banana's DNA that you extracted from this experiment. So, all right, I'm gonna try one more to give you a better visual. All right. I think this will give you a little better visual, but all right, can you see that? All right, that is what you're looking for. So that is it for the details of the experiment. And now we're going to move on to the recap of what we just learned.
So recap, at this point, you have already mixed the salt, water, and Dawn detergent in a glass or small bowl and set this mixture aside. The funnel lined with cheesecloth is in your glass tube side down. You added about 3 tablespoons of the extraction liquid, salt, water, and detergent in the bag with the mashed fruit. Then, you poured the strawberry mixture from the bag into the funnel with the glass under it. Your glass is about 1/4 full. You tilted the test tube and poured the rubbing alcohol down the side. And finally, my favorite part, you saw the DNA separate between the layers. What did you do next? You put your bamboo skewer in the test tube where the alcohol and strawberry layers meet. And the whitest stringy stuff is DNA from the nucleus of the fruit cells. You have completed your experiment.
All right, everyone, that concludes our DNA extraction experiment. Thank you all so much for coming and hanging out with us here at STEMCon. I hope you are able to take something away from what you learned today and apply it in your future science classes. Be sure to take everything that you used today, clean out all the dishes you used, and put away everything that you used from around your house so that your mom doesn't get upset. Let's be honest, she knows where everything is. All right, so that is all for now. We look forward to seeing you at future STEMCon events.